Hello everyone, I'm Hyunju postdoctor from 성균관 University. I am very honored to speak here. Today, I will talk about the subject of the arginine methylation as a key post-translational modification in skeletal muscle homeostasis. The maintenance of the skeletal muscle mass and physiology is essential for health. Skeletal muscle is the largest organ in human body, comprising about 50% of the body weight. Aging, overnutrition, cachexia, or chronic inflammation result in the debilitating the muscle quality. Of course, we can improve our body muscle mass and quality through doing the exercise, calorie restriction, and eating some supplement. Sarcopenia, which means the muscle wasting, is recently given the new disease code in the US. There are many reasons leading to muscle wasting. Among them, our group studying the proteostasis for muscle maintenance. Muscles are affected by the protein synthesis and degradation to maintain. Increasing AKT-mTOR signaling pathway stimulate the protein synthesis through the ribosome and supporting muscle rebuilding. In contrast, activation and nuclear translocation of FOXO3 via starvation or disused muscle can turn on the protein degradation via autophagy and ubiquitin proteasomal degradation pathway. Not surprisingly, defect within this proteostasis system result in the muscle wasting. Protein arginine methyltransferase is one of the post-translational modifier that regulate the histone or non-histone substrate. Among 9 identified PRMTs, PRMT1 regulate the asymmetric dimethylation on substrate, and PRMT1 is widely expressed and functions as a predominant of PRMTs, account for 80%. The deletion of PRMT1 causes early embryonic lethality before the embryonic day 6.5. Previous studies have shown that PRMTs is very important for the glucose metabolism and muscle regeneration. PRMT1, 4, 5, and 6 involved in regulation of glucose metabolism through the regulation of FOXO1 or crab-binding protein. Furthermore, PRMT1, 4, 5, and 7 are critical for stem cell function in muscle regeneration. However, we don't know the direct role of the PRMTs in muscle homeostasis. So our group focused on the role of the PRMT1 during skeletal muscle remodeling. Using skeletal muscle-specific PRMT1 knockout mice, we examined the function and downstream target of PRMT1. So first, PRMT1 fluxed mice were bred with the MLC-3 mice. The body weight of the 6 months old PRMT1 knockout mice exhibit did not show any difference with the control mice, and the body composition by using the NMR analyzer reveals that limb mass was significantly reduced in knockout mice. However, the fat mass were enhanced in knockout mice compared to control mice. And CAS and TA and EDL muscles were significantly reduced in knockout mice. However, soleus mass was not altered in the knockout mice compared to control mice. To examine the detailed analysis for the muscle phenotype, we did the immunostaining. PRMT1 knockout mice exhibit the decline the type 2 myofiber size. In qRT-PCR analysis reveals that muscle atrophy-associated protein atropine and MURF1 levels were significantly elevated in PRMT1 knockout muscle. And then we examine the muscle function through the grip strength and isometric force. First of all, grip strength was significantly reduced in PRMT1 knockout mice. Furthermore, isometric twitch and tetanic force were declined in the PRMT1 knockout mice. These data collectively suggest that PRMT1 deficiency causes muscle wasting and weakness. To determine the molecular mechanism of the muscle atrophy via PRMT1 deletion, we examine the expression change of the PRMT1 in different energy state. So TA muscle are harvested from 3 different energy states: normally fed, fasted for 16 hours, and refed 6 hours. Phospho-EMPK and LC3-2 levels were significantly enhanced in fasted muscle, and then they decreased in refed muscle. Interestingly, PRMT1 levels were reduced in fasted muscle and then enhanced in refed muscle. This data suggests a potential role for PRMT1 in control of energy sensing pathway. To define the energy sensing pathway of the PRMT1 deficiency, control and PRMT1 knockout mice were fasted for 6 hours and then treated with the insulin for 10 minutes. Without insulin treatment, phospho-AMPK and LC3-2 levels were significantly elevated in PRMT knockout muscle. With insulin treatment condition, phospho-AKT levels were not altered in the PRMT1 knockout muscle. However, the phospho-AMPK and LC3-2 levels were stayed higher in PRMT1 knockout muscle. This data suggests PRMT1 deficient muscle exhibit the elevated catabolic pathway with increased autophagy. FOXO is key regulator for induction of the autophagy and protein degradation linked with the muscle atrophy. So we examine the expression change of the FOXOs. As you can see, FOXO1 levels were not altered in PRMT1 knockout muscle. However, FOXO3 is strikingly elevated in PRMT1 knockout muscle. Previous studies have shown that arginine methylation of FOXO1 via PRMT1 regulate the hepatic gluconeogenesis and apoptosis. Since FOXO3 contains a consensus site like FOXO1, so we examine the methylation assay via starvation condition or other PRMTs. As expected, asymmetric methylation was increased in energy-depleted state. Interestingly, PRMT1 levels were reduced in energy-absent state. So we hypothesized that PRMT1 does not directly methylate the FOXO3. So we examine the other PRMT levels in PRMT1 knockout muscle. Also, several PRMT levels were altered in the PRMT1 knockout muscle. PRMT6, mRNA, and protein levels were enhanced in the PRMT1 knockout mice. So we further define the relationship between PRMT1 and PRMT6 on regulation of FOXO3. We conduct a nuclear fractionation in control and PRMT1 knockout C2C12 cell. FOXO3 and PRMT6 protein levels were elevated in the nucleus and in total lysate, which correlated well with the elevated levels of MURF1. PRMT6 overexpressed cells shows enhances the levels of the FOXO3 in the nucleus as well as total lysate. So this data suggests PRMT1 deficiency enhances the activation of FOXO3 is likely mediated by the PRMT6. And then we analyzed whether PRMT6 induces the FOXO3 via methylation. So we carried out the immunoprecipitation with FOXO3 along with the PRMTs. PRMT1 is bind with the FOXO3. However, it failed to methylate it. However, the PRMT6 is bind with the PRMT6, and then they induce this methylation on arginine residue of FOXO3. Finally, we to determine the potential methylation site of FOXO3 via PRMT6, we carried out the mass spectrometry analysis. So 3 potential methylation sites were identified. And then we generate the arginine to lysine mutant. So as you can see, FOXO3, the methylation of FOXO3 wild type and 2A18 enhances were enhanced. However, the arginine 188 and 249 failed to methylation. Furthermore, downstream target, including the atropine and MURF1 levels, were reduced in arginine 188, 188 and 249 expressed cells. In conclusion, PRMT1 deficiency in muscle enhances the levels of PRMT6, and then PRMT6 methylate the FOXO3 at arginine residue of 188, 249, and then they thereby enhancing the autophagy and protein degradation. So taken together, PRMT1 is a key regulator for PRMT6 FOXO3 axis in control of the autophagy and protein degradation underlying muscle maintenance. So finally, it is our lab member. I always thanks to Professor Jong-Seung her guidance. We collaborate with the Professor Seung-Hae Gu from Korea University. Thank you for listening.